Okay, this is the video portion of my exam 3 for cell biology. So I'm going to show you the drawings I did for mitosis and meiosis. Okay, so I drew this stuff out to talk about mitosis, but before mitosis happens, interphase occurs, which is the growth phase, and in the S phase of interphase, the nuclear DNA doubles. And so we can see two centrosomes with centriole pairs, the plasma membrane, the nucleolus, the nuclear envelope, and then condensed chromatin. And then when we start mitosis, we start with prophase, in which individual chromosomes condense to the point of being discrete visible objects. So we can see our chromosomes here, and they consist of two chromatids, and then they're attached tightly at the centromere. And then in animal cells, the nucleoli disperses as chromosomes condense. But in plant cells, nucleoli may remain as discrete entities. They may undergo partial disruption, or they may disappear entirely. But so you can see the nucleolus disappearing here. And this is a structure called the aster. And then these are the microtubules forming mitotic spindle. And then in prometaphase, it's marked by fragmentation of the membranes of the nuclear envelope. So you can see the fragments of the nuclear envelope. And then breakdown of the nuclear envelope allows spindle microtubules to make contact with chromosomes, as you can see. And the spindle microtubules are destined to attach at the centromere. And the kinetochore is a structure that attaches paired chromatids to spindle microtubules. So the green structure is the kinetochore. And then this is the spindle pole, and then this is atrial microtubules. And then we go on to metaphase. And in metaphase, the fully condensed chromosomes align at the metaphase plate. So you can see this is the metaphase plate, and we can see all the chromosomes lined up nicely on it. And then this is the kinetochore microtubule because it's connected directly to the kinetochore. This is a polar microtubule because it's connected to the outside. And then this as a whole is the mitotic spindle. And then we go on to anaphase. And in anaphase, two sister chromatids of each chromosome separate and begin moving toward opposite poles. And so in anaphase A, the chromosomes are pulled by their centromere first. And then in anaphase B, the poles move away from each other. So you can see it kind of looks like the cell's like elongating. And then in telophase and cytokinesis, the chromosomes uncoil into chromatin. So we can see the chromosomes decondensing. And nucleoli develop at nucleolar organizing sites on DNA. So we can see our nucleoli forming. And spindle disassembles, so you can see there's no more spindle. And nuclear envelopes form around the two groups of daughter chromosomes. So we're starting to get our nuclear envelopes back. And then cytokinesis occurs, and you can see the cleavage furrow of where that's going to happen. So I've drawn these to show what happens in meiosis 1 and meiosis 2. So we start with meiosis 1, and that starts with prophase 1. So that starts with leptotene, in which the duplicated chromosomes condense. So you can see maternal and paternal DNA, whichever you want to represent either one. And then in zygotene, there's continued condensation that makes individual chromosomes distinguishable, and homologous chromosomes become closely paired via synapsis. So you can see the synaptonemal complex being formed between these homologous chromosomes. So this bivalent is forming, and this synaptonemal complex forms in between to aid in further processes and the next steps. And so each bivalent has 4 chromatids, and so 2 from each chromosome. And then we go on to pachytene, where crossing over occurs, and that's marked by a dramatic compacting process, reducing each chromosome to less than a quarter of its original length. That was kind of hard to show in this drawing with the little space I had. And then, so synapsis is complete in pachytene. And so that synapsis is also called homologous recombination, and that's responsible for a good part of where the genetic variability comes from, because it's combining maternal and paternal DNA kind of randomly where they cross over. So you have a different combination in each different bivalent. And then in diploidine, the chiasma is visible, and so that shows where they crossed over. And then, so homologous chromosomes of each bivalent begin to separate from each other near the centromere, and 2 chromosomes of each homologous pair remain attached by the chiasmata. So it's hard to see in this drawing, but it would be right there. Visual evidence is provided that crossing over occurred by exchanged DNA segments. So you can tell that the maternal and paternal DNAs crossed over. And then the last part of prophase 1 is dikinesis, where the chromosomes recondense to maximally compacted state. The centromeres of homologous chromosomes separate further, and chiasmata become only remaining attachments between homologs. And the nucleoli disappear, the spindle forms, and nuclear envelope breaks down. So that's hard to kind of show in this drawing because there was limited space, but you can tell that they're more separated. Then we go on to metaphase 1. And in metaphase 1, bivalents attach by their kinetochores to spindle microtubules and migrate to the spindle equator. You can kind of see how they're lined up in the middle at the spindle equator. And then each bivalent has 4 chromatids, so 4 kinetochores are present, and they're orientated randomly in terms of maternal and paternal bivalents. And then this contributes to genetic variability again in anaphase 1, where members of each pair of homologous chromosomes separate from each other and start migrating toward opposite spindle poles, pulled by their respective kinetochore microtubules. And each pole remains a haploid set of chromosomes. And in this, independent assortment occurs, which is the other source of genetic variability, because there's a random combination of maternal and paternal chromosomes being pulled to each side. And then in telophase 1 and cytokinesis, then sometimes the nuclear envelopes form around the chromosomes before cytokinesis ensues, generating 2 haploid cells whose chromosomes consist of sister chromatids. Chromosomes typically don't decondense before meiosis II. So then we go over to meiosis 2, which is a little more similar to mitosis. So in prophase 2, it's very brief and much like the mitotic prophase 1. Well, the only mitotic prophase. The only difference is there's obviously different combinations of maternal and paternal DNA. But you see the chromosomes condensed, and they're each composed of 2 sister chromatids, and the centrosomes are separating from each other and moving towards opposite sides of the nucleus. And then you can see the centrioles. And then in metaphase 2, it resembles mitotic, but it has half as many chromosomes present at the spindle equator. And the kinetochores of sister chromatids face in opposite directions, allowing sister chromatids to separate and move as new daughter chromosomes to the opposite spindle poles during anaphase 2. And then in anaphase 2, it's very similar to mitotic anaphase, and the sister chromatids are pulled to opposite poles, and then the poles start to move away from each other. And then in telophase and cytokinesis, it's also very similar to mitotic telophase and cytokinesis. And the chromosomes uncoil back into chromatin, nucleoli begin to develop at the nucleolar organizing sites on DNA. Spindle disassembles and nuclear envelopes form around the 2 groups of daughter chromosomes. And then the final result would be 4 haploid daughter cells. Okay, so I wanted to emphasize what I said about genetic variability in meiosis. So the first place where genetic variability can occur is in prophase 1, and this is when crossing over occurs. And so the maternal and paternal DNA can cross over on the chromosomes. And so that gives opportunity for variable different combinations of maternal and paternal DNA. And then the other place where genetic variability occurs is in anaphase 1 of meiosis, and independent assortment occurs, and so they're randomly pulled to different sides of the poles, and then this ends up having all genetically different haploid cells. So that just there's all kinds of assortments that could be made from that, so that makes there be more genetic variability. Thanks for watching.